Today we're going to be talking about the process called the cell cycle. The cell cycle is a process in which one cell, called a parent cell, divides into two cells, which are called daughter cells. Now the first thing I'd like us to do is just take a look at this short video clip. Which shows the process of a cell dividing. In this video you can see one cell dividing to become two cells. And there are actually two general events going on. First, the nucleus has to divide, and second, the cytoplasm has to divide. In this next video, we're actually looking under a microscope at a culture of some nerve cells or neurons. And these neurons are in the process of dividing. Now it's hard to see altogether what's going on, but if you follow any one of these nerve cells, you can see them get larger and larger and then eventually split into two. And again, what's happening is that one cell is dividing first its nucleus and then its cytoplasm to become two daughter cells. Now there are a lot of functions of cell division in a living organism. Probably the most obvious is the function of growth and development. So any organism that begins as a small embryo eventually has to become a much larger adult organism. For example, in this slide we can see an embryo that has two cells in it, right here, and that happens to be a sand dollar, embryo. And that embryo will become eventually an adult sand dollar, like the type you might find on a beach somewhere. Obviously there's a big difference between these two cells and the millions and millions of cells that compose the adult sand dollar. So a lot of growth had to have taken place, and development in the sense that cells had to change their roles as they moved from the embryonic form into the adult form. Another very common function of cell division is tissue repair and renewal. There are two pictures on this slide. The first one shows a smear of blood underneath a microscope. And there are all different cell types in here. There are red blood cells, which appear in pink. And then there are various white blood cells, which appear in the blue and purple colors. Now this white blood cell in the center, which is a lymphocyte, is dividing. Now the nucleus, the purple area, has already divided, and now the cytoplasm, the blue area, is divided. So we started off with one cell, and now we're going to end up with two daughter cells. Of course, the bone marrow is a place where cell division occurs quite frequently. Over on the right-hand side, we can see an image that depicts the layers of skin that you and I have in our body. And what they're trying to show us is that there's been a wound on this piece of skin. So here we can see the outer layers of skin, the dead cells, if you will, the flattened dead cells that make up the surface of our skin. And then we have the subcutaneous tissue underneath, and then the layer of the dermis. And you can see blood vessels running through this dermal layer. When you cut yourself or lacerate your skin, it's these dermal vessels that cause the bleeding. And the first thing that's going to happen in the process of wound healing is that that bleeding will be stopped by a blood clot. Once that happens, then the skin has to repair itself. And that means that all of the cells throughout the epidermis and the subcutaneous and the dermal layer have to start dividing in order to repair and replace this damaged tissue. The third very common function of cell division is asexual reproduction. So here we're talking about an organism that doesn't have a distinct male and female and doesn't use gametes in order to produce a new offspring. Here we're talking about organisms that reproduce asexually, and they too have to go through a process of dividing their nucleus and then dividing their cytoplasm. So we can talk about cell division in terms of the cell cycle. And all that term refers to is all of the events that lead up to the division of a parent cell into two daughter cells. And again, that's going to include a nuclear division and then a cytoplasmic division. Now, when the cytoplasm divides, we give this a name. We call this cytokinesis. The cyto portion of this word refers to cell, and kinesis refers to movement. So altogether, cytokinesis means cell movement. And it got this name because when early scientists were looking through microscopes at dividing cells, they could see the cell physically moving as cytokinesis was taking place. Now, there's a lot of things going on during the cell cycle that you can't see under the microscope. And these are the processes which prepare the cell for nuclear division and cytoplasmic division. These include doubling the volume of the cell, doubling all of the contents of the cell, and doubling the chromosomes. Everything has to be doubled or copied before it can be split or divided into each of the new daughter cells. When we look at the cell cycle, we can think of it in stages. Just like any cycle, it's going to continue around and around like the hands travel around a clock. And we can divide it into very distinct stages based on the events that are occurring at that one time. We call these stages the G1, the S, the G2, and the M. G1 stands for the first gap, or gap 1, and this is a time during the cell cycle when cell contents are doubled. This is also a time when the cell monitors this doubling process and makes sure that there are no errors. The S phase is a time called synthesis, and this refers to DNA synthesis. This is the time when chromosomes become doubled. Then we go into a second gap phase where again cell contents is continuing to be doubled and the cell is continuing to make sure that this doubling process is occurring without errors. The last stage is called the M stage, and this refers to nuclear division. We call it M phase, but in different cells that M can stand for different things. This is the stage where the nucleus is going to divide. And if we're talking about a nucleus in a somatic or a body cell, then M stands for mitosis. But if we're talking about the M phase in a gamete, then the M stands for meiosis. They're two different processes. They have different steps involved, and they lead to differing results. But both of the stages, mitosis and meiosis, are a nuclear division. There is a fifth stage to the cell cycle that doesn't fit around the clock face, but it is still important. We call it G0. This is a stage that a cell can go into when it stops dividing. So this is how we might draw the cell cycle. And as you can see, it's presented as a circle like most cycles are. And we can divide that circle up into the various stages. You can see on the right-hand side the G1 or the gap 1. Then that arrowhead moves into the S phase where DNA is synthesized. That's S. Then we go into the G2 or the second gap stage. And then we go into M. And in this slide, the M is standing for mitosis. That tells us that we're talking about a cell cycle in a somatic cell. The other thing to note in this slide is that it says eukaryotic cell cycle. And that's because this cell cycle with these stages would only be found in a eukaryotic cell. Notice too that there's an arrow that leaves this cycle. This would be the G0, and that's for cells that leave the cycle and stop dividing. Now, most of the things that go on during the cell cycle are going to happen without us actually seeing them underneath the microscope. The G1 stage, the S, and the G2 stage are all invisible underneath the light microscope. The things that are being doubled during those stages, the contents of the cytoplasm, the organelles, and even the DNA itself in the chromosomes, those things are not visible to our eye underneath a light microscope. For that reason, we often group them together, the first three stages, and call that part the interphase. So you'll often hear or read about the cell cycle in terms of interphase and M. The reason M gets left out is because M is visible. Remember, we said that during nuclear division, when a cell is dividing, chromosomes actually condense. They get shorter, they get fatter, and they become visible underneath the light microscope with appropriate staining. After we go through interphase, G1, S, and G2, and through the M stage, mitosis in this example, then we're ready for cytokinesis, which is when the cytoplasm itself divides. And when we're talking about an animal cell, we're going to physically see the membrane of that cell pinching off in the middle so that we then have two brand new cells. So here's another depiction of the cell cycle drawn out. You can see G1, you can see S, G2, and then M. And in this picture, they've also drawn in cytokinesis as a little sliver of time after the M phase. Notice that after M, there are now two arrows branching off because now we have two daughter cells. There's also a G0 depiction over here for a cell that is arresting or leaving the cell cycle. So during the first gap, we have cytoplasmic contents being doubled. Everything except the chromosomes would fall into this category. And so when you look down here, you still have one cell after G1, and nothing much looks different. Then we go through the S phase, which is the synthesis phase, where the DNA gets doubled. And at the end of this stage, if we were able to see it, we would see a double copy of all the chromosomes inside the cell nucleus. In the G2, or the second gap stage, we're also duplicating the cytoplasmic contents and checking to make sure that there aren't any errors in everything that's happened so far. You can imagine that during interphase, G1, S, and G2, there are opportunities for mistakes to be made. And anytime we talk about a living cell, there's always opportunities for mistakes. Now, a cell has a lot invested in making sure that the daughter cells it produces are perfect. And so for that reason, it will put the time and the energy and the resources into double checking the things that have been doubled during G1, S, and G2. If we were to look at the cell at the end of G2, not a whole lot would change. The cytoplasmic contents would be completely doubled, but it would be something we couldn't see. And again, we would have two full copies of DNA, but it would be something that we couldn't see. So now we're ready to move into M phase, and in this slide, the M is standing for mitosis. This would be the process of nuclear division. One copy of chromosomes would go into one nucleus, one copy of chromosomes would go into the other nucleus. So at the end of mitosis, we have one cell that has two nuclei in it. Very, very strange. But that leads directly into cytokinesis, where that one cell divides into two, and we now have two daughter cells. Now I wanted to take a minute here and talk a little bit about how your textbook draws the events of mitosis, the M phase. Remember that normally a human cell is going to have 46 chromosomes in the nucleus, or 23 pairs. Now, it would be difficult to draw a nucleus that had 23 pair of chromosomes in it without students getting confused as to what's happening inside that nucleus. And for that reason, your textbook chooses to use a cell for its depictions of mitosis that only has 4 chromosomes, or 2 pair of chromosomes. And the way your book does it is they show 2 long chromosomes and 2 short chromosomes. So in other words, there's a pair of long chromosomes and a pair of short chromosomes. And when they get replicated or copied during the S phase, it looks something like this. 2 long chromosomes, 2 long chromosomes, 2 short chromosomes, and 2 short chromosomes. So keep in mind when you're using your textbook to study that we're looking at the cell of an imaginary organism that only has 4 chromosomes inside of it. And for that reason, when we get to the end of mitosis in this imaginary organism, our daughter cells, our brand new daughter cells, are only going to have 4 chromosomes inside of them. Okay, so we've studied the cell cycle quite a bit over the past many decades, and we've studied it in different cell types because the length of the cell cycle varies depending on the type of cell that you're looking at. It takes some cells a relatively short amount of time to divide, while other cells take a much longer time to divide. Interestingly enough, the S phase and the M phase stay relatively constant no matter what cell type you're talking about. So, for example, intestinal cells are one of the cell types that divide very rapidly. Intestinal cells are the cells that line the cavity of your intestine, and they take quite a beating as digestion takes place and absorption takes place. And so they tend to slough off and get damaged and so on and so on. So new cells have to be rapidly produced so they can replace the old cells. And that process takes about 12 hours in a human being. So about every 12 hours, the lining of your intestine is replaced. Now, the S and the M stages stay relatively constant. And in the G2 stage essentially stays the same. It's really the G1 stage that takes the big hit. The G1 stage is shortened, 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 shortened until it's essentially eliminated in these cells. It doesn't mean that the cytoplasmic contents aren't copied. They are. It's just done in a much faster amount of time. It turns out that copying DNA during S and dividing the nucleus during M take a certain amount of time. You cannot speed up S and M very much. Now, if we look on the other end of the scale, we can find cells that either divide very slowly or don't divide at all. If we look at mature nerve cells, for example, and mature muscle cells in a human being, these are cells that don't normally divide once they're in place in an adult organism. So we would say that these cells have arrested, that they've left the G1 and gone into G0. They've stopped dividing. They're in the G0 phase. Now you notice I have a little asterisk up here, and that's because you and I in 2014 are living in the age of stem cell science. And it turns out that while normal mature nerve cells and normal mature muscle cells do not divide, it is possible now for us to harvest what are called stem cells from these tissues, and sometimes we can turn them on and ask them to go back in time and start dividing. Now, the other thing to note is that the length of the cell cycle changes under different conditions. And what I mean by that is that there are times during the life of an organism when the cell cycle is faster or slower. The growth period for organisms is a time when the cell cycle tends to be very fast versus the adult stage where most of the cell cycle speed slows down. And then there are also instances in the life of an organism when something happens that causes the cell cycle to suddenly change its speed. For example, in a human being, liver cells are on a very slow cell cycle. Once your liver is fully formed and in place, a typical liver cell lasts about a year. So we don't need to replace them any faster than a 12-month cycle. But if you damage a human liver, and I mean significantly, the cell cycle will suddenly speed up, and it can speed up to a 24 to 48 hour cycle, which is pretty amazing. It's been shown that in humans who damage their liver, let's say in a car accident, you can lose up to 70% of your liver that way. And that 30% piece that's left behind will kick into gear into a 24 to 48 hour cell cycle. And over the next several weeks and months, all of the remaining cells will be replaced with brand new cells. Now, the other good example of the changing length of the cell cycle is in other organisms, such as yeast and various bacteria. It turns out that these organisms are very, very sensitive to their environment, which makes sense. We're talking about single-celled organisms that are surrounded by conditions that change. And so these organisms will change the length of their cell cycle depending on whether or not the conditions are good to produce new cells. If there's plenty of nutrients around and if the environment is favorable, the cell cycle length tends to increase. Whereas if the environment is particularly hostile at that moment, then those cells tend to not divide or